I'd like to introduce David Kasahara. So David Kasahara actually is from our university, right? So he had his bachelor degree in pharmacy and biochemistry at the University of São Paulo. He went down to the United States. He had his master here at the University of São Paulo, and he went down to Boston and to the United States for his doctorate and postdoctorate. He went to University of Vermont and to USP-Harvard as well. So now he's currently a research associate at the Department of Environmental Health at the USP-Harvard. So thank you for coming.
Thank you for inviting me for this talk and to opportunity to. Thank you for inviting me for this talk, opportunity to interact with this cohort of the students. That will be very exciting. Number one, before I start to talk about my science, we have, let me talk a little bit about our school. So you guys, we end up coming to the School of Public Health, which the new dean, Dean Frank, he's from Mexico. He was the former minister of the public health in Mexico, and also he was a member of the World Health Organization, which you guys know very well. And he brought some idea. They say, well, as a school, we should have a motto, a mission of the school. And this is the mission: powerful ideas for healthy worlds. So basically, in the school, you guys will be involved, like John Golaski showed some data about air pollution. So how we can change this world to someplace better, someplace healthier. I was very excited in our lab. I work for Dr. Stephanie Shore since 2008. And in our lab, we do pretty much stuff that seems like completely out from the School of Public Health. We do a lot of lung physiology, and as well as we study pulmonary immunology in our lab using animal models. And in the past few years, we had the opportunity to host our lab for several Brazilians. Lucas Chen, I don't know if you guys know, he being our lab. Leandro being our lab. Fernanda Ninin, Felipe Lazar, and right now Luisa Ribeiro. And all fantastic students. They are doing very well. And most of our students had the opportunity, as John showed, 40 publications from the students. Some of the students, they get the first author paper. In our lab, most of the students get at least 2 or 3 co-authorship paper. So we work hard, but also they had a great time, and they had some fun, in the lab. This morning I will talk something about rho kinase in allergic airways disease, which is a little bit off what we do in the lab. In our lab, actually, we focus more in the question about obesity and asthma. So we try to understand the relationship between the both, either using looking the immunological response to air pollution, obesity, and how they induce obese people to get asthma symptoms. Or right now we are exploring something cool, which is a microbiome. So pretty much the next student is coming over will be involved in those 2 projects. This is pretty much over. This project is about to be published soon. So let me tell you what I'm going to cover this morning. I'll talk a little bit about allergic asthma, which I'm assuming that at this point you guys know very well after had some class with the Paulo Saldiva and Burns there in the pathology. I'll talk about this molecule. I don't know if you guys learn in the cell biology, rho kinase. I will cover slightly about rho kinase and lymphocytes, and what is the role of the ROCK2 and ROCK1 in allergic inflammation. Asthma, as you can see here, this is a normal airways, and this is asthmatic airways. So in asthmatic airways, you're going to see accumulation of the mucus in the lumen. Inflamed airways, usually a lot of eosinophils and the lymphocytes. Constriction of the muscle cells, and this result in narrow airway, so it will be very hard to breathe. So this is extremely oversimplification of the mechanism of asthma. Allergen interact with the disease, the dendritic cells. They message CD4 cells, T lymphocytes, which polarize, expand to type 2 lymphocytes. And the type 2 is because they start to express type 2 cytokines or pro-inflammatory molecules such as IL-13 and IL-5. IL-5 actually is involved in recruitment of eosinophils, and IL-13 is involved in inducing airways of cells to become hyperresponsive, and also they make small muscle cells to release eutaxin, which also is known as the most potent chemotactic factor for eosinophils. So let me move a little bit about the Rho kinase. Rho kinase is a protein of the 160 kilodaltons. Is a serine threonine kinase, so they phosphorylate targets for phosphorylated bunch proteins. And you can find Rho kinase in 2 flavors. You can find two isoforms, ROC2 and ROC1. Both have around 82% homology in the kinase domain. So this means that they pretty much they share similar targets. However, you have 2 regions here that the homology is less than desirable. So it's about 55% homology in the co domain and 68% for the plexstatin homology domain. This means that probably the difference between ROC1 and ROC2 is the place of the cells that they are expressed, and also in the different they're expressed slightly amounts in different tissues. Here, for instance, this is if you go to NIH website, you can put any gene you want, and then they're able to show the distribution of the expression of the genes in different tissues. This is the mice actually. You can pretty much see that Rho kinase 1 and 2 is ubiquitous expressed. So pretty much virtually every single cell in the body express ROC2 and ROC1 in the mice, as well in the humans. So this is very relevant target. So what is the function of the ROC? So let me show you back. I forgot to tell you that here in the coil domain, this is a place where the Rho kinases get activated. One of the main way to get activated is by binding Rho A here in this part of the molecule. So Rho A bind to the ROC and phosphorylated many different hundreds of proteins, different proteins. And the phosphorylation of those proteins induce contraction, focal adhesion. You're gonna, if you come to the HS page, you're gonna hear a lot about this, especially those who work for Jeff Fredberg. Cell motility, cytokinesis, proliferation, and cell cycle control. So what can activate Rho A, or what can induce Rho A? A lot of pro-inflammatory marker, and in asthma, probably IL-13, S1P, bradykinin, those can activate Rho A to induce this. So the question now is Rho A, is ROC is activating asthma? So this is one of the example which we took a mice, we exposed to ova, which is allergens. It's a classical model because it's number one, you cannot induce asthma in mice. So then we use mice as a way to understand more molecular mechanism. And the people use ovalbumin as a way to induce allergic response. And in this case here, we observe phosphorylation myosin binding subunit, which is one subunit of the myosin light chain phosphatase, which is very specific. There's a specific phosphorylation site for ROC1 and ROC2. So this is very specific marker of the activation ROC. And we can see here that after you challenge mice with OVA, there is a significant increase, actually quite robust increase of the phosphorylation of the myosin bind subunit. So this means that, yes, in the allergic model, you see activation of the ROC. And then someone did experiment which they look at inflammation and they look at airway hyperresponsiveness. In this case, he exposed mice OVA, he challenged, and he treated with a drug, fasudil, which block ROC kinase. So ROC kinase is an inhibitor, so they don't work. And we see here there's significant decrease of the inflammation measured by number of eosinophils. As well, this was flex event, which is a system you measure pulmonary function, and that is a decreased response to methacholine in the mice that you block ROC kinase. So next, we try to correlate this for another phenomenon. So in asthma, if you deplete CD4 cells, this is a model which you challenge mice some antigen, and then you use the antibody. Actually, by the way, this using antibody to deplete cells or to deplete the cytokine is become more popular in terms of the therapy. So I think the next generation, or you guys will start to prescribe a lot of those biologicals, which is antibody to neutralize cytokines or cells. And here, in this experiment, Marsha Wilcarps, she used anti-CD4 and see a significant decrease in lymphocytes and eosinophils, as well as attenuation of airway hyperresponsiveness, which looks like similar to the previous slide observed in the ROC kinase inhibition. Yes?
Which means delta PT.
Yeah, so this she measure what they call response. So this is how much they respond to methacholine. It's like the PC 200.
Okay, so this is delta pressure?
Yeah, it's a pressure. It's a pressure over time, yes.
Pressure over time. So it probably represents the increase in pressure during mechanical ventilations?
Yes.
Unit of time.
It's the nature of time, yes.
Sorry.
Oh, that's fine. So now our question here, based on the both slides, is there a role in the ROC kinase in the lymphocytes, or because it's quite suggestive. So we know from the literature that ROC kinase is required for development of the cells. So without ROC kinase, the cell is not able to develop fully. They're not able to polarize. This is important for chemotaxis attachment. Without ROC kinase, basically, they in vitro, at least, they don't get attached, they don't move. Activation and apoptosis. So these are some data proving that. So here, this was a Mingzhu in our lab. We published in 2011. We took cells, T cells from mice. We stimulated in vitro with the OVA, and we look at proliferation of the cells by measure this radioactive probe. And we treat mice before that with the another ROC kinase inhibitor we call Y27632. So here we see actually a significant decrease of proliferation of lymphocytes when you decrease ROC activation. And in this study, we observe also by decreasing, blocking ROC kinase, you see decrease of the chemotaxis, so the cells are able to move, as well as the function. So here was again Taki. We see using fasudil, we decrease the ROC kinase, and we see less IL-5 and less IL-13 in bronchoalveolar lavage. Okay, so then our hypothesis in this study is the Rho kinase in CD4 cells required for allergic airway inflammation. To answer this hypothesis, if this hypothesis is true, our prediction is that by attenuating ROC in mice, we'll be able to see that some of the allergic inflammation will be pretty much will be gone or will be significantly attenuated. And also, I did an experiment which adopted transfer mice with the ROC. The mice that they have a ROC in CD4 cells in mice that is ROC insufficient, and then I look if I'm able to reverse the attenuation of the response. So this is the experiment. In our lab, you guys are going to work a lot with mice. So if you be very, don't be afraid to work with mice if you come to our lab. That's what we do all the time. We used a black 6 background, the wild type, a ROC1 and ROC2 plus minus, so they only express 50% of ROC1 or 50% of ROC2. The reason we use a heterozygous, that homozygous, well, number one, they survive you're going to get one knockouts in the 64, 65 liter, so they die, before you're able to use them. This is the classical way to induce allergic response. You do OVA plus aluminum hydroxide times 0, 14, and we challenge day 28, 29, 30, and we measure pulmonary function inflammation 48 hours after last challenge. However, on top of that, we decide to add cells from another animal. We used OT2 mice, which is a black 6 background. In OT2 mice, they have a normal expression of the ROC1, ROC2, so they're normal, except that these mice express a transgene for a TCR receptor, which you guys probably know, the T-cell receptor, and that T-cell receptor is specific for ovalbumin. So this means that you don't have to sensitize those cells. And then we isolate CD4 cells and we inject back to the animal, and then we challenge to see if we are able to restore some of the feature probably impaired by ROC. So this is how I isolate the cells. We took a spleen. We used a system with the antibodies, which binds to mast cells, neutrophils, eosinophils, monocytes, and basically by binding that, I'm able to do a negative selection of CD4 cells using magnet. Part of CD4 cells, I did a surface staining, and then I measure how much enrichment I got it. Before enrichment, the spleen, you can get around 25% of the cells as CD4 positive, and after enrichment, my enriched population is around 80% the CD4 positive cell. Another way to do the enrichment will be using cell sorting in the flow cytometer. However, the issue with the cell sorting that you have antibody already bind to those cells, so they might decrease a little bit their function. I will talk a little bit about the ROC2. We did this experiment with ROC2, and we observed that in ROC2 we see attenuation of the lymphocytes in bronchoalveolar lavage, which we measure. If you go to the clinicians, you put the stuff there to measure, take the cells from the lungs. In the mice, we intubate, we throw the saline, take it out, we count the cells. And then when we adopted transfer CD4 cells, we restore the number of the lymphocytes. Obviously, we put a lot more CD4 cells in the body. Then we look at IL-13, which is one of the marker that the cells get activated. and then we observe that in ROCK2 plus minus, we have a less activation of those cells. We put back CD4 cells, and we restore the amount of IL-13. Okay? Now we look at IL-5 as well. Let's say, well, there might be IL-5 will be fine, you know. And actually we see attenuation of the IL-5. We put back CD4 cells, we restore, we rescue the type 2 response. Then you can ask, well, okay, that's nice, but do you think that this might decrease eosinophils? And if this is able to restore eosinophils, because we know that the Rho kinase can decrease migration of eosinophils in the tissue. So if in fact, you know, what you the issue is that there is a decreased Rho kinase in eosinophils, you see less migration, right? So in fact, we see less eosinophils in the tissue here, you know, after in the ROCK2 plus minus. So maybe by putting back CD4 cells, I will not be able to restore those eosinophils. In fact, I did. Seems like the eosinophils fine, you know, they're able to migrate. So this showed that probably the issue, in fact, is in the CD4 cells. So now we can measure eotaxin, so we can think that, well, in that case, maybe you need the lymphocytes to produce IL-13, so this means that you have eotaxin in those animals. In this paper, actually, Taki showed the same thing, less eotaxin after the block of the ROCK with fasudil, and we observe, yes, we do have a less eotaxin. So let me restore, you know, with the CD4 cells. So maybe I'm able to restore eotaxin, which I did not. So it seems like small muscle cells getting affected by the ROCK as well in this model. We can look at airway hyperresponsiveness. Okay, we can, you know, block ROCK2. Maybe they only affect inflammation, but nothing happens in terms of the mechanics, right? Actually, this is a nice airway hyperresponsiveness signal. So this is the G, which measure small airways. We see hyperresponse. Flex event, yeah, that's correct. We measure response to methacholine, and in the ROCK2 plus minus, we see pretty much no response or similar response. Looks like a wild type PBS unchallenged. Okay, so I can give you back ROCK2, say, well, we restore IL-13, you know, so yeah, the mechanical response will be back. Actually, they didn't come back. So this means that the ROCK is very important also for a smooth cell contraction. We did some tracheal experiment. This is combining ROCK1 and ROCK2 plus minus. We see attenuation of the maximal response in the tissue contractility. This was an isometric contractility assay. So right now, this interim conclusion, I can conclude that target ROCK2 can attenuate the T-cell dependent inflammation, and it seems like, or my data showed that ROCK2 in no CD4 cell is the key for developing airway hyperresponsiveness. So I can try to do some experiment with those CD4 cells. I can decrease the ROCK only in CD4 cells by using a system that mice that they express Cre recombinase. We crossbreed a mice here that have a promoter for transcription factor that induce CD4. And this is very specific. So once you start, you create your Cre recombinase gene, you put it here very close to the CD4 promoter. So once you activated this, they start to produce cree recombinase, and basically they look for the flank LOXP site in the ROCK2 gene, and then basically they cut them, you know, and the animal, they are deleted. So basically they don't express ROCK2 in that particular cells that express CD4 protein. So now we use the same ovalbumin model. My control was ROCK2 flox as well as CD4 cree, and here's my experiment. So we look at the same marker, lymphocytes L13. There's no change. Nothing happens, you know, when they took it out the ROCK2 in those cells. Eosinophils, pretty much the same. There's no attenuation of number of the eosinophils when they took ROCK2 in the specific CD4 cells. And we look at, you know, airway hyperresponse. They still respond the same as the control. So this means that, you know, probably the ROCK2 is not acting in specific CD4 cells, but in someplace else. Our guess is endothelial cells, which requires for transmigration of the lymphocytes from the bloodstream to the tissue. Would be another complicated experiment to do. In this experiment also we use the flow cytometer to look at different cellular population. And interesting, nothing changing when you look at IL type 2 response. So like T cells with the intracellular staining for IL-13, similar. However, this was striking. We see less IL-17 cells in this model. However, this model is not IL-17 dependent. We use this more as a way how to prove that in these animals they have in fact, you know, less ROCK2 activation in those CD4 cells. Let me skip this conclusion and go quicker for ROCK1 model. So the other isoform is the ROCK1. In fact, you know, in the ROCK1 we see less lymphocytes. Looks like on the ROCK2 we put the CD4 cells to restore lymphocytes, but however there's no change. So this is a significant, and pretty much there's no difference between ROCK1 and wild type in terms of the IL-13 as well. But when we put back CD4 cells, they have a huge increase of IL-13, IL-5, similar. Eosinophils, there are no change in eosinophils, so this seems like it's not being affected by ROCK1. Eotaxin, we able to restore eotaxin. Here, huge expression of eotaxin, which corroborates with the idea that IL-13 induce eotaxin. And then we look mechanical response. Okay, like ROCK2, we see attenuation of airway hyperresponsiveness, and then when add CD4 cells, we restore the airway hyperresponsiveness. So this is cool, you know. So in the previous one, we couldn't restore, and this we can restore HR. So we can conclude here that allergic airway inflammation was not impacted by ROCK1 insufficiency. However, the allergy-induced HR requires ROCK1, pretty much looks like in the ROCK2, and adoptive transfer CD4 cells was able to reverse this effect in ROCK1 plus minus. I can conclude this morning that ROCK2 plays a central role in allergy-induced inflammation. Allergy-induced response are independent of ROCK2 insufficient CD4 cells, and ROCK1 plays a role in allergy-induced HR. This is probably one of the most important slides in this talk this morning. So if you treat asthmatic, all right, you go to the clinic, you know, well, let's see what's the repertoire of the drugs I can use. So far, those that are most currently used for asthma, you will see a lot of bronchodilator, either short or long-acting bronchodilators. corticosteroids, which will decrease inflammation, anti-leukotriene, some people love to use that, I don't know why. You can decrease IgE, and thermoplasty is extremely efficient in some cases. However, my data suggests that, probably we could add another one. We could add a drug which is act on the Rho kinase, especially in the Rho kinase 2. The drug right now is on they are in the phase 1 or phase 2 clinical trial so far. None of them is being used for asthma. However, the KD025 here is being proposed to be used for like lung fibrosis. But this is there's a potential once they solve some issues about side effect and how you deliver the drug specifically to certain areas of the lung. Another important slide is this one, is my acknowledgement. I would like to thank Stephanie Shore, which was my mentor, which if someone decides to come over, you guys will have opportunity to meet her. Joe Matos helped with the flow cytometer, but then there's a lot of Brazilian students. So Luisa, which she was running some of my qPCR in this cohort. I didn't show any qPCR data here, however. Fernanda Ninin, she did a lot of a challenge. She worked with the mice, although she said, I hate work with the animal, but she did some work. I convinced her to do some. Philippe, did a great job with us. Lucas Chen and Leandro, so be involved also in this project. This is the guy who created mice, and the financial support from NIH. I will open for questions. Thank you very much.
So before sandwich, you have
differential expression, quantitative expression between ROC1 and ROC2, between the lymphocytes, CD4, airways, muscle, and then in the models.
The lymphocytes, this guy, Jim, he looked at, but he never published the data. He's holding the data. He say, "This is cool. I'm not going to show the data." So he never showed it. But someone did it, and we don't know actually the answer for that.
Quite collaborative, isn't it?
It is. Yeah, he's holding the data, but seems like they might have, differential expression. Actually, if you go back here, Paulo, this paper by Biswas. In this model, he took the lymphocytes and then he threw a bunch of cytokines to polarize the cells, right? Seems like in terms of expression, they're the same. They have a similar level of expression. However, the activation might be different.
Well, but I'm talking about the since ROC1 was deficient and mice deficient of ROC1. Have a decrease after responsiveness and then you restore.
Yes.
So you can reverse mechanically.
Yes, that's true.
ROC2 is not. So there are differences in expression.
Gene expression probably not, because
Do you have any marker of the immunohistochemical marker?
No, we don't. We don't have any marker. So I regret that I don't have that. But I have lungs. I can go back, and run the Western or.
Very interesting. When you cross CR mice with rock, you became a croc. So you transform the modification. So this is the power observation because
Thais?
Do you have experience with Alba's mind models? Why are you using Alba?
Oh, yes. Actually, I forgot to tell you that this guy here, go back here, he started to do some experiment, how does might. But, number one, it's a very expensive model, number one. Because we used a high purified house dust mite from Greer, so we buy from that. And we want to do, our plan was doing a chronic experiment because we want to look at remodeling of the airways. And you needed, you have to give a house dust mite intranasal, a relatively high amount of house dust mite for every day for, I think my memory was 6 weeks every day.
I bought it for $300. It's not so expensive.
Well, one of those $300 vial, you're gonna run out in, if you do a cohort of the 6 animals per group, which we have here 3 groups, rock 1, rock 2, and the wild type. We run out in probably 2 weeks, 2-3 weeks. Because I have the protocol that they use a very, very high quantity of the house dust mite. And we use the one that is like they have as less as possible endotoxin. Yeah, but we try, and our issue was the model. We couldn't get a consistent model, so then we decide to leave for as a next project. But you bring a good point.
So do you have any other question from students or no? So David, maybe some students got afraid from the immunology things, and what do you say? Yeah, maybe, right? Yeah. So what do you say that these students can expect from the lab in terms of knowing more about immunology? What do you think is the most difficult part, or what they can expect from the lab in terms of learning?
Oh, okay.
Molecular data?
I think the immunology is the easy one. Although that some of them will be a little bit afraid, I will tell you why it's easy. Because in our office we talk about immunology 24/7. Because I'm glad that I'm not immunologist, but 2 of my colleagues, Joe Matthews and Jeff Brand, they're both immunologists. And we try to teach our students a little bit about the recall immunology. And now the key word, in terms of immunology is we talk a lot about innate lymphoid cells. So,
Do you manage to mark these cells already?
we use a flow. So basically, for looking at cells, we do all the markers, right? And then we look at those cells that doesn't have those markers.
Or you do a specific combination?
We use it CD4, CD8, CD120, something. But those, to do what they call lineage negative.
Yeah.
So, and then
By exclusion.
by exclusion, yes. And then we use a c-kit or ST2, to look at ILC2s, for instance. Or we use ROR gamma T to look at ILC3s. So we have to do some kind of intracellular staining. Otherwise, it will be impossible to differentiate. But don't be afraid. You guys will learn that. It's like a good idea to take what they call the little Abbas. Just a quick, the read. If you come to the lab. But
I can give some lectures on immunology if they want before you go, because it's what I studied. So before I should I can see I give you some lectures so that you get there.
yeah, but the thing is, we're not working anymore with the allergic model. I'm the only one work allergic model. You guys will work a lot with the what they call innate immunology.
And that's the good point, is to learn immunology in a contextualized way, and when it's your field of work, you see that it's very different for having 2 or 3 lectures of some group of cytokines or cells that you are not looking forward for what the hell this will work. So
Yes.
when you are working with it, you learn it the same as we do with statistics and so on.
David, in your opinion, what kind of, in the line of what Professor Thais mentioned, what would be the pre-training So you're talking about remodeling.
Oh, yes, yes. For the students before go there, right?
Yeah.
So I recommend, if you have a chance to read or a quick, revision. Biochemistry, everyone hates biochemistry, but biochemistry is extremely important. Cell biology, because that will be applicable to all the labs. And then you can talk to your mentor to look at specifics. Like some labs do more immunology, like our lab do, like lung physiology, immunology. So the time the students arrive in our lab, we give the what they call the West, right? And then also we mentor, to learn immunology. But cell biology and physiology, cell biology and biochemistry.
About theoretical information. I would like to know whether there's any practical, for instance. I know, John, so basics of pathology or immunology or depends on, because you're talking about remodeling. Remodeling will probably measure collagen.
Yes.
It's muscle repair or something like this.
Well, one of the things that we come to appreciate is that the students really have some theoretical knowledge, but they don't have much practical bench experience. And so one of the things that we're planning to do this year, and Dr. Verrier has done this for a while, where he had a group of students make videos of the things that they do in terms of methodology used in the lab. And so actually we're planning with this year's group of students to make a whole series of basic laboratory videos that actually should be available to the students before they leave Brazil, so that not only should you have a chance to look through these videos, you may have a chance to actually go through them and have sessions where you talk to the alumni and they can answer questions about the videos for you. And then when you come, you'll not only see the videos again, but you'll then get specific instructions for how those videos relate to your lab. So we're hoping to have a didactic package for the students before they leave Brazil, so that as you go through December and your Christmas vacation, you can be looking at these videos as something that you'll be learning and using once you get to Boston. So we're planning that kind of a program for the coming year.
I suggested to put a soundtrack of Christmas songs in the video.
Well, you see, the idea from the video comes from Liana and Andre's group when they made this great video of Boston and things in the lab, just with really good music too. Yeah, started shipping up to Boston, and this video made me think about the possibility of this year's group really making these training videos in a very effective way. So we're going to be working on that. That will help the students as they come to the lab to have a better idea of some very simple methodologies that you will come to use on a regular basis, and that often gets ignored because it's just not practical to teach you that in medical school. But once you come to the lab, it's going to be important for you to know it, and so that you're going to have several sources from which to learn. So hopefully these will work out.
And also, in terms of theory, I think that ideally, is to read a few papers, 2 or 3 papers where the lab been done, the past year, because most likely those type of projects that you'll be involved, there's a follow-up from those papers.
Okay, but can you send us a representative list
Oh yes, if you once you know the students be selected to each mentor, because each mentor have a different type of the work. So you have a mentor be work more into mechanical, you know, stress, you know, and okay. So once we. Oh, I can try to coordinate that. You know, once we select the students, I can talk to the PIs, gather the paper, assemble a package, and send it to you guys. And you guys are welcome to email me or email probably your more close mentor, because each one will be assigned probably a postdoc to mentor you. So then you can start interacting before arriving there. And ask a question. Oh, I don't understand what this means. You know, this molecule or this method or.
Yeah, so just in terms of the alumni things, we have been doing this, but to each PI every year. But this is great to have a way to institutionalize it, right? This is what we want. So, but we depend from your input. So from the members that come this year for 2015, if you want to have, you know, previous information, talk to the students, things that we can do that we've already thought about. We can have meetings with the previous students from the alumni with you that were selected. We can have the professors from here, as Thais is mentioning, welcoming you to their labs here at FMUSP to get you prepared. We can have papers sent from the professors to you for you to get prepared, and we can have videos and training. So these are all things that we can do all together. So, but we depend from your input, your interest in doing those things. But we, from the alumni, we can do it very well. So we have a coffee break here.